do you see next gen RNA approaches sort of offering any distinct advantages, right, from a supply chain sourcing standpoint, compared to, right, a linear mRNA product? And the delivery, I will admit, is a challenge, right? We're still, LNPs are a whole category in and of themselves, and who knows what else we're cooking up, right? So when it comes to the drug substance here, right, from your RNA perspective, April, do you have any comments on, does this simplify things at all from a supply chain management as we get more advanced?
I definitely agree with Greg around like circular RNA. Like if we can get that advanced, it takes away the capping, it takes away the tailing, it automatically stabilizes itself. Like there's potentially some huge benefits there.
Yeah.
Slightly different manufacturing approach probably, but it's probably feasible and manageable. And I think once we understand the applicability of circular RNA to vaccines, to gene editing tool delivery, I think there's some real possibilities there. Self-amplifying RNA, I think, has the benefit probably to the patient. I would envisage, self-amplifying RNA theoretically can go in at a lower dose.
Yes.
Theoretically can elicit, less of a reactogenicity and things in the patient. So I think there's some real benefits there. But from a manufacturing perspective, it's going to be tougher. These are much bigger. They encode, 2 or 3 extra proteins to do the amplification part. The plasmids are bigger, the RNA is bigger, they're harder to keep intact and purify. So that's going to be tough. I've seen issues with that at manufacturers at least a couple years ago. They're just bigger molecules. The other thing with self-amplifying mRNA, depending on like what genes you're actually, or what proteins are doing the amplification, if you're using alpha virus genes, trucking those around across country borders can be a big barrier because they're the pathogen element, they're select agent, even though they're not replication competent by a long shot. And in many countries, they're viewed as GMOs, so importation can be quite challenging. And then some countries really regulate even the clinical use or administration of GMOs like that. So, But the patient benefits probably make it worth the while.
Yeah, and we have actually seen some success, right? We have a self-amplified RNA vaccine out there in the world now, thanks to the Japanese, right, regulatory agencies. So we're starting to see a little bit of movement, but I agree with you. They're big boys, right? We're working with something. And also just in general, you brought up a really good point too about even just the logistics, right, of getting the supplies that you need to manage them and getting them across borders, right? As I think Greg, you and I have talked about this. This is everything's sort of disconnected, right? We're all just sort of bringing pieces together and tying them together from a supply chain standpoint. I think I used the metaphor cat's cradle in a recent article, right? So strings everywhere. There's strings everywhere.
When you got a great product and you're all set to do a clinical trial, and the Australian OGTD, the GMO organization, they're like, well, you need to like get it a permit, and that's going to take like almost a year, and then
a year.
How these like little details can really handicap the program.
Greg, what were you going to say?
2 points that April made that are, I think, important. I don't want to be lost on the audience's, the potential for lower dosing and maybe less frequent dosing in the therapeutic setting that these new modalities may offer, and also the stability. So that could translate into, just lower scale, lower cost of goods overall, and better thermostability in general. So those certainly would be advantages on the CMC side that we can tap into. But a long way to go clinically and ultimately commercially for these. But let's not forget about that.
Yeah, that's a really big one. Cost of goods. Hello. Are there any? Let's see some poll questions here, or some audience questions. I'd seen one come through here on the cost and supply challenges you're facing in novel modality manufacturing. And do you use relatively the same processes? So it looks like they single out self-amplifying and circular RNAs here. I know we just talked a little bit about that, but do either of you have any other comments in terms of, cost or supply challenges? April, maybe we can start with you, and Greg, if you have any additional.
Cost and supply challenges related to the mRNA versus the circular RNA?
It looks like what cost and supply chain do you face in novel modality manufacturing? So it would be in the modality, in the manufacturing or making of self-amplifying or circular RNA?
I mean, I think circular RNA actually resolves some of the issues because you don't, like circular RNA self-splices itself. So there's way less enzymes involved. So that actually helps fix some of the problems, I think. Self-amplifying mRNA, or yeah, it depends on what resin you settled on that'll actually purify it properly.
Okay.
Hopefully you can get that resin.
Yeah. Greg, any
Yeah,
other?
and I think April pointed out earlier that some of the manufacturing challenges in terms of like what self-amplified, dealing with a bigger mRNA, more susceptible to degradation, harder to purify in general. So that's all going to lead to some cost disadvantages, whether it's because of lower yields, more resin need, more purification steps. You know, so, but of course that's offset by what I said earlier about lower dosing and you know, less materials that you might need to synthesize. So you know, in terms of my experience in getting into large scale clinical supplies, it's really with linear. So I can't say on balance where we'll land, but I think those are the, you know, the factors.
Yeah.